Welcome to the world of Biodyssea. We aim to provide videos that are as accessible to as many people as possible, in attempting to answer to three main questions: what are our origins, our nature, and our future? To begin with, the point is to respond to a simple interrogation. What are the components that make up life? This first part aims to reveal you what are the bricks involved in building life and without which our world would be mineral and inert. Let's start with some overviews. All living beings on Earth are composed of one or several base units called cell, which is actually the smallest living unit. Thus, in adult human, it is estimated that there are about 10 to the 14, that is to say 100,000 billion cells. Of course, these cells are not completely identical and present a huge diversity of size and structure. It seems obvious that skin cells and muscle cells present different properties and features. In human, 200 different types of cells can be distinguished. Strikingly, all living multicellular organisms, including fish, reptilian, mammals, but also mushrooms and plants, derive from one single cell, which through a species-specific genetic program and some individual characteristics will give rise to all the other ones and make the living beings as we know them today. Depending on the complexity of these different organisms, cells' number, size and composition are changing. Two different types of cells can be distinguished. On one hand, there is the prokaryote cells, like bacteria, which are unicellular, about 1 micron, and contain as much as necessary to survive. On the other hand, eukaryote cells that make up mushrooms, plants and animals present more complex structures. They are most of the time multicellular, but it also exists unicellular organisms and present a huge variety of size and structure. With sizes of 10 to 100 microns, eukaryotes are therefore 2,000-fold more voluminous than prokaryotes. Let's take a closer look at the differences between them. Although both of them present an external membrane, also called plasma membrane, delimiting the so-called cytoplasma or internal cellular space, and allowing a selective permeability with the environment, such as diffusion of water and other molecules, the distinction between eukaryotes and prokaryotes, in addition to the difference in size, is made by the compartmentalization existing only in eukaryotes. Hence, while genetic information encoded by DNA is found free in the unique prokaryotic compartment, in eukaryotic cells, DNA is enclosed within the nucleus, the biggest organelle of the cell, delimited by two nuclear membranes that isolate it from the rest of the cell. Nucleus also includes one or several nucleolus. Many other organelles, also delimited by membranes, can be found in eukaryotes, such as endoplasmic reticulum, forming an interconnected network of flattened, membrane-enclosed sacs or tubule-like structures around the nucleus, and the entire cell is also crossed and supported by an important network of cytoskeleton, crucial for the cell shape and mobility. Eukaryotes also present a mitochondrial network of particular significance for cell metabolism, a Golgi apparatus comprising a series of 5 to 8 cup-shaped membrane-covered sacs that look something like a stack of deflated balloons, and finally some lysosomes and peroxisomes, spherical organelles with specialist functions. It has to be noted that plant cells in particular present in addition chloroplasts, organelles in charge of photosynthesis, vacuole, and a rigid cellular wall. Last but not least, you should have noticed the presence of flagella and bacteria that I have chosen to not represent in eukaryotes. In fact, these flagella that permit the cells to move in the environment also exist in eukaryotes, but not systematically. I won't spend too much time on discussing about the functions of these organelles that deserve to be highlighted in a dedicated video, but for those who are not familiar with biology, eukaryotic cell functionment can be compared to a small city, which not only manages its self-production to survive and to be autonomous, but also its import-export system to find its place in its environment. At the molecular level, both eukaryotic and prokaryotic cells are composed of the same components, mineral and organic. Let's take the example of the human body composition expressed as approximate percentages of the body weight. Water represents more than 60%, mineral salts 5%. Then the rest of the body is represented by organic molecules which are specific of living beings and in which we distinguish 4 major classes of distinct biological macromolecules: proteins for around 18%, lipids representing 10%, then 3.5% of nucleic acids including DNA, and finally 1.5% of carbohydrates. Let's get to the heart of the matter by starting with sugar. For a better understanding of the particularities of each biomolecule, we have to go deeper into chemistry. Okay, just a bit. So, carbohydrates, also called saccharides, are, as their name indicates, hydrates of carbons. That means association of one carbon atom and one water molecule. Of course, you remember that water, designated by the chemical formula H2O, is formed by one oxygen and two hydrogen atoms. To be called carbohydrates, molecules should be composed of a chain of at least 3 carbons. One should wear a carbonyl group, in other words, a carbon linked by a double bond to an oxygen, and if this carbonyl group is located at the end of the carbon chain, it is an aldehyde function, otherwise, in the middle of the chain, it's a ketone function. So, one carbonyl group and at least 2 hydroxyl groups. In other words, all the remaining carbons are linked to an oxygen bind to an hydrogen, an hydroxyl group. As a carbon atom is making 4 bonds in total, thus, in addition to these carbonyl and hydroxyl groups, it is connected also to additional hydrogens. We basically separated saccharides in 3 categories: monosaccharides, disaccharides, and polysaccharides. Let's start with the simplest ones, the monosaccharides, also termed sugar, which are the smallest unit of carbohydrates. 4 physicochemical features are used to distinguish them. First, depending on the carbonyl group, we will make a distinction between aldose, presenting an aldehyde group, and ketose, presenting a ketone group. You got the point. Just have a look on these 2 monosaccharides with the same number of carbons. With the aldehyde function, the glyceraldehyde is an aldose, as opposed to the dihydroxyacetone, which is a ketose. The same chemical formula, but different names and different properties. Likewise, depending on the number of carbons in the monosaccharide, we present sugars as trioses, tetroses, pentoses, etc. Due to the fact that carbohydrates contain multiple chiral centers, many isomers are possible, including enantiomers. Chiral center is linked to the presence of an asymmetric carbon atom, linked to 4 different groups or atoms. The spatial arrangement of these groups around the carbon is kind of stuck, meaning that, like in this example here, if you interchange 2 groups, like the blue and the brown for instance, this leads to 2 different molecules, which are non-superimposable mirror images of one another, and they are called enantiomers, either D or L, and they also present different properties. To be simpler and easier, it will be like compare your right hand with your left one. An example of enantiomers is the D and L isomers of glyceraldehydes, as shown here. But nature prefers D isomers because they are those which are most naturally occurring. As many carbons can compose a chain of monosaccharides, many of them can be asymmetric, leading to the corresponding number of enantiomers possible. Last characteristic, pentoses and hexoses can cyclase in aqueous solution, as the aldehyde and ketone group can react with an hydroxyl group of one of the distal carbons, forming a cyclic hemiacetal. A 6-member ring is called furanose, whereas a 5-member ring is termed furanose. Thus, among furanose are fructose and ribose. Ribose which plays a crucial role in nucleic acid composition, and among furanose we can find glucose and galactose. This leads to a new annotation to distinguish anomers alpha or beta. To illustrate this physico-chemical part, let's consider an example well known to all of us, the glucose. From a chemical formula recapitulating the atomic composition of the whole molecule, we can build a semi-developed formula with our nice chain of carbon. There is 6 carbons, it's a hexose. The initial carbon binds only 1 oxygen and 1 hydrogen, consequence of a carbon-oxygen double bond, therefore it's an aldose. So as a feature projection, we can have a better idea of the chemical structure, and we can better appreciate the configuration of asymmetric carbons. With this projection, you can differentiate the different enantiomers. Here you can distinguish the L-glucose from the D-glucose. The D-glucose is predominant in nature, and as I said before, can cyclase in aqueous solution, like our blood. This cyclization gives rise to 2 anomers, alpha and beta, which are in balance with each other, and can interconvert by mutarotation from one to another. As I just mentioned it, the D-glucose, a hexose and an aldose, is the most important simple sugar in nature. Glucose is one of the primary molecules which serves as energy sources for plants and animals. Another famous monosaccharide is the D-fructose, or fruit sugar, a ketonic hexose found in honey, fruits, and most root vegetables. Last example, the D-galactose, both hexose and aldose, like the glucose, is, as you may know, present in milk and milk products. One major function of monosaccharides is to be used as a form of energy for the organisms that consume them. Glucose, for instance, the major energy source, is metabolized within cells to create fuel. In the presence of oxygen, glucose breaks down into carbon dioxide and water, and energy is released as a byproduct. Glucose is a product of photosynthesis, and plants obtain energy from glucose through respiration. Humans acquire glucose from food, and the body transforms this monosaccharide into energy. Monosaccharides solo the transport of energy thanks to their solubility in water. Thus, in animals, it is through the blood that they can be distributed to all cells, and finally, they also serve as building material to elaborate more complex molecules like polysaccharides. Let's see now the next group, the disaccharides. They consist of two monosaccharides linked by a covalent bond. We find in this category the saccharose, our table sugar, composed of one fructose and one glucose. We also found the lactose, the most important sugar present in mammals' milk, composed of one galactose and one glucose, and finally the maltose, composed of 2 units of glucose. It's interesting to note that saccharose is the transport forms of sugar in most of the plants. That is why our table sugar derives from sugar crops, in particular beet and cane. Last, only monosaccharides and disaccharides have a sweetening power. Polysaccharides are tasteless. Finally, let's conclude this first part with polysaccharides or glucosides, composed of several monosaccharides linked by covalent bond. They could be either polymers of the same monosaccharides or polymers of a mix of different monosaccharides. Polysaccharide chains can be straight or branched, and they can be composed of hundreds to thousands of units. In nature, polymers of glucose are predominant. So let's speak about the major polysaccharides and their functions. Polymers are involved in 3 main biological functions. First, they are mainly used as food storage, like the starch in plants and glycogen in mammals. Both of them are glucose polymers. The difference between them? The kind of covalent bond between glucose units, and in particular of which carbons is involved in the covalent bond. Polysaccharides play also a structural role for the maintenance of cell shape, like cellulose and pectin in plants, or chitin in hard external skeleton of insects and crustaceans. Thus, you should know that paper and cotton are made of pure cellulose. Finally, polysaccharides have an important function in cell signaling, notably owing to the glucosaminoglycans, another kind of straight polymer of modified saccharides. These glucosaminoglycans are important components of extracellular matrix and conjunctive tissues. We are now coming to the end of this first part. I hope you have enjoyed it, and keep in mind that carbohydrates are fundamental to living beings. For my part, I look forward to seeing you in the next episode. See you soon.